Polymerase chain reaction would require a template and a lot of other components. The template DNA molecules, which are long molecules within which we are interested in a specific small region, maybe a gene of interest or some other region. We would like to amplify that region. So we start by adding this DNA template and a lot of other things together in a small reaction mix. And we put all of them in a tube. So the polymerase is the one enzyme that has a key role. Primers have a different key role. And of course, we do need a lot of free nucleotides and buffer, etc. The reaction mix will be subjected to heating and cooling cycles, about 30 of them. And then at the end of about 2 hours after 30 cycles, we would like to test if the reaction has worked. That means I would expect about 1 billion copies of the target molecule. How do I test that? By electrophoretic analysis. Now let's see, when I said 30 cycles, how each cycle works. The first cycle requires denaturation of the DNA by heating at 94 degrees centigrade, and then the separated DNA strands would then be annealing to the primers like this, forward primer and reverse primer. This marks the border of the PCR product eventually. So primer designing is very crucial. If we don't design primers, then a lot of things can go wrong. So there are certain videos I have made if you want a quick introduction in simple terms about primer designing associated principles. The primer annealing happens at 58 to 65 degrees centigrade. And the next step, the third step, is polymerization, where the enzyme is active at 72 degrees centigrade approximately, and it will polymerize, and you get this in the end of first cycle. Now the 2 new molecules that are formed in this cycle would form as a template in the next cycle, where the primer comes and binds and polymerization happens like this on both the strands. You note that at the end of the second cycle, we have for the first time 2 short single-stranded molecules produced, which are of the actual size we are interested in. Now in the third cycle, they serve as template and we get double-stranded DNA molecules for the first time of the target region. They will, amplify now. They will be produced in exponential quantities in the following cycles. And at the end of 30 cycles, we will get billion copies of these molecules, whereas others could be there, but they are in small quantity. It's essentially a pure DNA solution that we now have in the PCR mix. A part of that solution will be taken and we perform electrophoresis. If we see the expected band at the right size, we know PCR has almost worked, but we need to confirm it by sequencing in most cases. So if you're a beginner in PCR, there are a lot of things that you should know. Some points I have listed here. You can pause the video and note them. But quickly to move on, I am also listing examples of basic research, including some of the latest ones such as CRISPR results, like what are the mutations if you want to test, we can use PCR. PCR amplicons can be produced for next generation sequencing, etc. There are also a lot of different applications where PCR is used even today, and there are technical variants that we should be aware of, as well as related techniques, for example LAMP, CRISPR typing PCR, droplet digital PCR. These are some of the new things that have come up in the recent years. Very useful, and though the beginners definitely should note them. Now my intention is to make a lot of such small videos, though I call them 2-minute videos. The first 2 minutes I try to cover the basic concept, which could be pretty complex concept, but I will try to tell in simple terms. I intend to produce a series of them. Hope you like this one, and if you like, please do mark like and share it with others and tell others so that other students and your friends could be benefited by this video. Thank you very much.